That you all enjoyed your tea. We're going to start off this session with a discussion about benefits and drawbacks about multicellularity.
Percentage 2 yosha ne shell hepatitis teles ne tandang asuge delaya tampote ngasu kare kutskin system balaya thapon manden chapa imbaina telaya ginsen tanchon kare yo metelaya into the giants.
And to start off this discussion, though, we actually don't need to focus on cells in particular. I have this question up here that says, why work together? And you'll discuss this in your groups, but you can think about this just as us, as humans. What are some of the benefits of working together?
Ta tanda de baina chitsuge de gangla thonsa shin te jiwa gangla ya ne thapon la soba yo ma res ne nyamli chigunu go jinsen ka res ra nyam te leka chigunu go jinsen ka res ote kola ya ne tude na ro ches de ngasu mila kuna de dus ngasu mi baina mi ta nyamri ge leka chigunu go jinsen ka res ote kola tude na ro ches. Ready?
Should we say like 1 minute? I always say 1 minute.
Kamachi esa. Kamachi rang la method de trade na lo esa.
Taking notes or anything, if you need, let us know.
Okay. Yeah, thank you. Like for the afternoon activity?
Yeah, in case if you need anything, just let us know.
Okay, thank you. Thank you for such great discussion. I think we'll work on coming back together.
Yeah, la so ta nyam te trade che yensa.
For this one, let's hear from 3 teams what your thoughts are. Just for the sake of time, I think we won't be able to hear from everybody, but let's hear from 3 teams who want to volunteer.
Ta tanda de la nga su yin chi tsu se sa che ge che to ne ro ka sum gang ne de gang la kare gong ge ya de la ya ne su su gong su so ko ke de yins ro ka sum
ane tang ngarang zo chupang ne ne dolong gari chong song sena tanda de nyamli chi go ge jimsen so de gar sena so keeping che deres ane labor da ne biology gang ne shena tanda ge nyog pon to zo nyamten di go de korong zo soni che ta korong zo rab shima de pe ge che ta ane nyamli chi ge ores ane nyamli kang tang kan ina so chu keeping chi go wa ti che de nyamli chi ge ores o te chong so.
So we have discussed about why work together for the common benefit, or actually for benefit. In the case of slime mold, we see that they come together for the survival and reproduction. So also for humans, we work together for our survival, for benefit, for the common benefit.
Okay, yeah, and that's a great connection that we're making too, right, between like what are the humans doing and then also an example of what we saw in the video.
So in addition to the survival and the reproduction, why we work together? Because if when we work together, we can achieve bigger goals.
Okay, yeah, this is another great idea that I think actually I heard from a couple of teams as well, that there are some things that will just not be possible for, it could be either one cell or one organism. It's not possible for them to do it by themselves. So that's a benefit of having everyone working together.
Jawa chenpo dritkere se tinga dus dristambala chetsu nane mambo chigdi kola song sra sungde shas ta chichi bayna ko thapong chi nare kansa cheta basikin dramatika tinda chonin dramatika tinda nyamli chiwala tine yan druya gi ne tang tang dus.
And then let's hear from one more.
tanda ni nganzogi nyamle chegi da keben ro de karishugila na ta chida nyamle che de gi gel sosola ni ta pendo da soba 지금 원래 펜도 니래 거라 당간저 니 니가로 요아체 당냠내 제와들 때 페나 차샤는 다나쇼게 간저 니 치소기에 니 계주디서 간저 장가서서 되는데 미치기 원에 근데 라야갈 가보라 당간저 냠내 제원도 잠발라 대다기 계주 망하러 지라야 돼 다딩간저 냠내기 뒤부러지 지기 요쇼
tatilethene susulaya garla imbana penthodus mabuz hakundus gorwe so when we walk together it benefit not only individual but also the group or whoever is in the community. For instance, when we walk together in the morning to list down the common features about living organisms, living things, as we walk together, we have all lot of lists we can come up together that helps the group as well as individuals within the group.
Yeah, great. And this is essentially, it's also just making me think of some of the ideas of diversity, right? Like in terms of coming up with these ideas, by having more of you working together, we were able to come up with a greater variety of the ideas. And as we've talked about in evolution classes, that's really beneficial to have that variation.
That and the nanshingi ro nyamte cheba imbana ko thamongi te la penthoba mase ane te kete yanchi nyamle chibi kapse yanchi na chopa chiyo ba imbaina ko na chokoranjin te pa imbana te le yanko karsugre kete chisu shopa lekate debi kapse nangla ya jajon na chopa yo pa imbana yanko thu ko pa imban te le ma to singro was tadanga su chingu na deshe jabi kapse imbaina yan na chokoranjin te ke chembi imbine ta te yan deshe ngores.
And these are just a few examples, so for humans, of benefits to working together. And these are mostly just examples of sort of some different chores that would happen around the house. But the point of this is getting at that there's all these different tasks that need to be accomplished. And so humans can help each other by dividing them up. And this is similar to what can happen in cells, is that the cells are then you can have this type of cell that's going to accomplish this task, and different type of cells will accomplish a different task.
Tata de gane ngatsu kare deshe jagore se stambalaya nyamli chi goe ge junsente kola deshe jagore stangatsu milan de taba imbana mela ya ta chimjang chikana ni chimle mambo yorwa mambo yoba de ngatsu imbana mi mando la ya ko lengeng go shawa japchi de te ane su lengge de pa imbana thamong gane le ke shimbu de ge ne tang te yong go yorwa tin na shinge thapon la ya ta ran jang go res thapon ri chi ke ane ko ge cheli te mando ri che pa imbana ne su ge thapon ri mando ge cheli mando che de thamate la nyamli ge ngu la yong stam bala ya thamong ge ngu yong stam bala ya chopi ngu yong stam bala ya le ka jenda chama rang jing de chi yorwa su ngune de bala ten de tan thamong ngune so chambu chi de ngu ting go res.
All right, now I have what might be a slightly more difficult question, and that is that I would like for you to again discuss in your groups why we might not want to work together. And so we can think about this though as just anything you imagine might be like a drawback of working together or challenges of working together. Anything, that just might make this like a little bit difficult when working together.
Ta danda de ka se chula ya tro shenda chi tang ge tampo de tang tes manda res ta ani ta nyamli ma chi na nyamli mi chi ge jinsen ka res ra ta nyamli mi chi ge jinsen ka res stambala ya ne samlu dus tang ro chis nyamli che ba imbana kange karyo res donlen chi go karyo skale khapo karyo res nyamli che ba de la ya chung karyo res ra ta de stambala ya ne nyamli ma chi ge ya jinsen karyo me te ko la ya tu de chiro chis tangi nyamli chi ge means se te la ya jinsen ko la ya de chiro chis tu de chiro chis.
Okay, you have it.
nganzo direseta nganzo gyobu dogana chibu juice 타르무 도고는 냠도 주세 담베디나 신신이라 나서 네 냠레 체나 디저 디스 툴라도도스 견직디 디레시라 니어데야 삼수 샴바질 카르들나 냠레 체이나 소소기 삼수 요나 지용 도미도스 라마시라민도레트
so so one reason we don't want to walk together is there's a saying that if you want to reach your goal fast, walk alone. If you want further, walk with others. So like that, if you want to achieve your goal and fast, you need to walk individually. And also sometimes with relating to that, sometimes if you walk together, sometimes your ideas cannot be implemented. So if you want your idea to be implemented, you need to walk along, not together with others.
Yes.
That's two reasons.
Okay, yes, there's two ideas there, and I heard the idea of sort of speed or efficiency came up in another group as well. So yeah, those are all, these would be good considerations for humans and also for the cells, right? If a cell needs to accomplish something very quickly, there could be some benefit to working on their own. And that's just, I guess, something good for us to keep in mind. These, if we talk about them for humans, then we'll also think about how they're related to cells.
다 단다 데레스 단다 치수 손바나 시젠다 지금 저 따라자 아트들랑 대샤스케 땡아서 단 조번조번 지구요 바이마나로와 미치바나 리카tin 조번조번 차고네 수수치 샤바는 조번 차기네 단다 삼성 연구레스 때나 신게 데데 양고 타폰랑라야 지 비저지시는 느기요레스 next one
네 랑올 lakba ranzuk shorwa rawan thokba ani ranzal mingomba
that the inchina is on the car set the ram ambushon yeah
losing personal skill, losing personal freedom, losing personal identity.
So if we walk together, we will lose our personal identity, personal skills, personal independence or freedom. Yes. One more? Do you want to hear from one more?
Yes, let's do one more.
ngazo ge dire thangbo de limited resources la ngazo resource di nyong thawa na ni group din de na so share la kala khabu cha jara so sene chu de no khojo changma la nyam de share na ani so la mandaw ka nge ja ani resource ki nyong thawa la tene ani dine gi nyam ju che na ani ka nge chik thi dus niwa de ngazo decision making ngazo thak chu de thani chik ba do re do di dawrish ani chik dikhara na ngazo dine gi nyam ru swela yang thusis tang gore energy tang damre chidi saman dilayang ke mangbo do this ani ngazo simjing gi dina tawa na ani simjing susu gi rigdi color breed chela kebel tang la thapsi tang gore ani khojo dine nyam de chidi sam suga tang ngazo do ina su rigdi kebel tang di thakso kala ga bo shwera to susu gi rigdi nyaza chela ani kangi thi dus
So one is depend on the resources, and the other is on the decision making. For instance, if you have a limited resource, then if you want to live together, it is really difficult. And also, if you work together, it uses a lot. It uses a lot of energy and resource. That's on the resource side, and also decision making. So if you have a lot of people with different ideas, it's really difficult to make a decision. In the case of human beings, if work together, how are you going to decide which individual will select it to reproduce for the offspring or keep the generations going? So so there is difficulty in the decision making as well.
Great, thank you. And I have sort of 2 points that I'd like to highlight from the two most recent comments that we got. So yes, the point that we had up here, this will become really important for when we talk about the differentiation of cells to become different cell types, because in that case, this would happen in a multicellular organism, but since those cells are now so specialized, they would not be able to just go out on their own and then live as an individual cell.
Tadanda dinane chesuge kicheshe chugnane ne to niti ko ya zu karde ta kicheshe gi yes chite kare system balaya dinane shepatire sta rawa ta mambu chilagu yes korwa ta dinane nga suge thapon la kicheshe pa bana thapon chirge bana thapon igwe chi gu yorwa thapon nga suge chagwe chi de ta thapon ti chele gi thapon la jirgu yorwa chagwe chi bi kapsirwa inbai na ya Chapon chagu machelaya, and Chapon Samalan Jutka Gungu, the Chapon Yorwako, Chagu Machi at the pig, Cheli Chapon Maturity in Bana, go children do that the chicken said the Omaris.
And then the third point that we had is a very good one about the complexity of organisms. So while benefits will sometimes come from that increasing complexity of a multicellular organism, then there is also that complexity can occasionally lead to some other challenges. And like in this example, we were talking about like decision making and things like that. It would relate to cells, and that what is benefiting one cell type might not have that same benefit for a different cell type.
Ta shendachi kare system balaya ngatsu nyam ta mampon stamba dela yang chi ka thachog ranjin ko kiche shig dus ngatsu ta jingwe te ko thapon mampo cha de ta nyongjon chenpo cha yong stamba balaya yang ko ge tila tene de ta yang ko neta mampo chi la yang ko gechen tre ta shichen tre tene da yong yor es ta picha sha pa in baina ndanga tsugu thapon mampo gi chingu chi cha pa in baina ta nyamle ngo chi cha pa in baina thapon khashi chi ke chele sigro da ko kepen shi chenpo ra ba ta thapon khashi chi ke chele ta te la ya kepen mara ba ge ne ta te yong yor es tin sta ba la ya ane ko thapon mangdu gi thapon mangden gi chingu ta nyongza chenpo drba na sing ko tsule kepen yor da ya te la na sing ya ko la chen da ya chi shi chi ngem ba tin na yor es.
And the main sort of big picture take-home message from all of these discussions are that from a biological standpoint, it's not considered to be inherently better for something to be unicellular or multicellular. This is all just going to be a result of the interactions of the organism and the environment. So it's always just talking about being suited for a particular environment. So all of those conditions that are included there, that's what's going to influence whether something unicellular or multicellular.
That and the jingle rupa geteni shape in Baina, Chitapon Changden, the Tapon Mande in Baina, G get on Junte Kitchens Tambalaya, Co Lenchig Ranjan, Nico, Kependembare set, and Lenchinico Chunta Dembar set, and Nitan the Deshajagmaris. That the Kari Latin Deshajagres Tambalaya, Captain Nathan Tula Tesgro, that Taponti, and Nico Tapon Chigden, Mande in Banaco, Nesagi, Coiti Latin Dete, Coitan Yamton Joachia Netan Chigongorwa. Dilatani dete ane ko ata kependu ane tela ya chondu se ane tante desi jagres.
All right, so here we are just revisiting these learning objectives since we're sort of using these as our guide and to keep us focused on sort of some of the main topics that we have.
Ta tanda dene roi ngasuge tudirchia ge joshi de ta chi khaseu ta ge chete de us gelakti 1 minute gelak ke kugude. 용거대 마이크나 용거대 전셔도 내가 야고 고대 야 the get the mic teach the mic is not working
Mine
properly.
is not working?
Yeah.
You just need it closer?
Dina 고대
Are you able to hear this now?
다 고대 it's better. Can you speak something?
Okay, are you able to hear this? Or maybe it just needs,
yes, that's good.
I think it just needs closer.
Yeah.
Okay.
Yeah. Yeah, I need to like listen like this.
Yeah, thank you for letting me know. Did you say this?
Yes, you go ahead with
Okay.
the objective.
So what we've done so far, we've talked about transparent teaching and the metacognitive reflection, and we just completed our discussion of the benefits and drawbacks of unicellularity versus multicellularity.
Tanda pertang asu kare tayo reses tambalaya chesen ansegul honsen triag triteptelaya and tela kepen tanchung kare yo pata ani tik jelaya asu ge rangi rantok ke samshe se chor asu su samlu thela samlu ta ya de ge kola ani desejare tin anshinge ni thapon chigden ta thapon mande la ya ni chenta kepen kare yo pa kola tudchi yores.
And now we are going to go through a bit of review of genes and cells, and this is going to be with the goal of, for you all, to do a combination of things you have learned about evolution and genetics, and you will be discussing in your groups and relating what you've learned about genetics with that peppered moth example from the evolution classes.
Tanda de kasanga sukar chingin senada chesuge rigzi rigpi kola ya chijang yorwa rigzi rigpi kola jang yore ane shenda nga suge penji rigpi kola ya jang yore penji rigpi kola jang stambala ya chimala chidwa chimala oddi kola jang yore sta tanda de kapsa karichi ya in sena rigzi rigpa te ta penji rigpa gi nane chimala ge ko penji ko ti ti ko jang patte ni cha parla ya ane drewa chiga chigorwa oddi kola tu de ins.
All right, so for cell theory, I'll just put this all up here. This is what we've already been talking about here, right? But it's a reminder, this is coming from part of cell theory, that living things are made of cells and they can be unicellular or multicellular.
Tande tapunga namjong ne chongbares ane ko chiden sangba tapunga trabares ane telaya tapung chiden na dukore tapung mandai na dukores.
And actually we'll highlight here just that this is saying in the multicellular organisms that the cells are doing different jobs, and this is going to be that specialization process that the cells go through, and we're going to talk about that more since that's very related to development of organisms.
Tata de jelaya olaya tapung mandai bana ko tapung rig mando mampuch ke tribures ti kiche gores ta dikarsena tapung ti ko chagui chebalatene de tapong ge ta chele gi ngulan jirgo yores ta dim bana ko tapong mandang gi jingui chi tribya ge ta ngu su ti chagores ti nga su jela yang ko tang sapong to cheyins.
All right, and then these are just to show you a few examples of the types of unicellular and multicellular organisms that there can be. I just always think it's very interesting to see these pictures, and some of them even look a little artistic. But there are amoeba, paramecium, this euglena, and also a diatom there. And then I believe that's a bacterium on the other side.
Ta dela tabai bana tapong chiden gi chingula tabai bana dela pi khaj ko sha de tapong chiden de phajtele amoeba thasin che paramecium che te ola euglena te soyang che tsjong de bana ane disha go simbu cha go bacteria simbu cha go yores.
For multicellular, these are organisms that we see all the time. Unicellular, usually we can't because they're so small, but just a few examples for completeness here. There can be animals, fungi, plants, variety of types of multicellular organisms.
Ta tapong mandai bai na ta dela kojo pi dita resta digita ta dita dau mampu yoresta dinan singe tapong chigden bai na namjin bai na ngasu chi ta shaja ta bai na thoya khabo res chongchong insa.
And I wanted to just do a brief reminder about mitosis here, since this is the process that is actually going to create multicellular organisms.
Da da de ka sembara ngasu tapong chagu kola ya this is jagins topat zupon ji ko kola ro ta de ka res na tapong chigden ne tapong mangdan la jirwi gi jurum tires.
And I have been informed that you all spent quite a bit of time working on this last year, so this is just to be a reminder of these 6 different phases that occur in mitosis, and then just a few reminders about the timing of the process. So this is saying that the cells can take about 2 hours to divide. However, this will depend on the cell type, so there's some variation. It's not always a set 2 hours.
Ta da de bai na ro tapong chagu na zupon ji ko tela ya nanye lo la chitsun zenda ya pu so de ta ya pu chi jang yores ta tanda de la chitsu transu ta jam yes ta nga su de ka sa tapong ji ko la ta bai na ko ga ramba ma so na thunti ro tukung la du yores ta de phaju de la ta bai na ko chitsu ka sa chung go yo pala so pares ta chirge bai na tapong chagu che la ya zupon ji je te la ya chitsu ni sung go yores ya yanchi tapong mando la tende de tan chi chanyum dinda chi go manda yang shenda yang chi so mando go ya yang yong yores.
We'll also take a quick look here just at the inside of the cell so that as we're talking about cells this week, that we all have it fresh in our minds what all is inside the cell.
Ahanda shendaji karta sena thapon la ya aru mampu yo pinane thapon ge nalo la ya tripcha kar yo pate tagis.
So here we'll kind of just do this as like a quick response, but this is going to be related to organelles. From your recollection, what are some things that you remember are included inside of cells?
Ahanda de jobo chesuge ya chi tola len naro che isra ta chesuge ngoma jamba la ya shisha ba na ni thapon nang la ya wanta kare yo res. Mitochondria. Did you hear anything?
Maybe we'll have to call out kind of one at a time. It's too hard, I think, when there's all of them.
Yeah.
Nurture tardin
Mitochondria.
ani ribotase
Ah.
ribosome
Okay.
tani DNA
Ah.
DNA tenen nang no DNA ora
DNA.
Cytoplasm. Jesu. Cytoplasm.
Lysosome.
Okay, so we've heard lysosomes, mitochondria.
Serna transfer.
Ribosome.
DNA. DNA.
Okay, and DNA.
mRNA.
Chromosomes. Chromosomes.
Chromosomes. Okay, right. So the DNA will be organized in chromosomes.
Ah.
Cell membrane.
Gogi. You call it Goji or Gogi? Gogi body.
Oh, Golgi bodies, yes. Yeah. Okay. Yeah, this is great. So good. We've got a quick review of some organelles here, and most of these are going to pop up here too. So keep in mind, the list that I have here is saying what all cells have. There are a few that are specific to like plants and animal cells.
That de la ya chesuge le ya puchen son swa. That de la ya ko yan thamon to yo pa khachta ya khashim ba na ya karsanga suge sochak ke thapon ta ane karsa session ke thapon la yan ko thamon to yo pa ta thamo mai me go su la yo pe gi yan khachin yo res.
So a few of these we mentioned and a few we didn't get to. So there's the genetic material that in many cell types would be found in the nucleus, although not all cells have that. We've got the cytoplasm and the cell membrane, which is going to go on the outside, and then the ribosomes that we've mentioned, which is where the proteins are made.
That is really by na tilayanga su DNA ta RNA yo bachero ane kanron yo patner wa tigila thatsi tigila thapung chmuche tigila ya ne robot has race.
Okay, there's some errors, but it's hard to see. Okay, we are now going to do a little bit more focus on talking about the plasma membrane and just talking about some of its properties and the way that it works. And I think this is something that you all also spent some time on last year, so this will just be another refresher.
Tandanga su thapung chmu gala jitagi yes ro de la ya ko chako karim bata shele karim bata gi yes de gala ye su ge nanye ya so ta jang yo res this tambala ye de la tensu do chi chak res.
And so this is going to be one of our first cases where we're really going to talk about the structure and the function. So I'm going to talk a little bit about the structure of the plasma membrane, and then that's really important for the function that the membrane is actually providing for the cell.
Ta tanda de ka sanga su karita gi yen senar wa ta chako ta shele go kola tampu chita gi yes ta tampu de bane chusar chimu de ge ne ko thapung ko thala yo ba tela tene de ta ko go pa kan ro chako kar yo pa ta tela tene de ta ko chele kar chi de yo mete ta yes.
The membrane will be found on the outside of all cell types, so whether it's an animal cell, plant cell, they all have the cell membrane. And as you'll probably recall, it has that phospholipid bilayer, so it's got the two-layer structure.
Ta tanda thapung ge chusar chimuti imba na ne socha ta zisheng ge thapung ni chala den ba res ni chala yo res ta ko thapung ge chimuti dbya de ko ramba ni den che ro wa ane ko inchikela phospholipid do wa omega ramba ni cheng ge dbyo res.
And then in terms of the job or the function of the plasma membrane, One of the major functions here is that it's going to just be controlling what's able to go in and out of the cell.
Ahata koge chele te sote roa jimuge chele sote ka resista la thapongge chelone nalo ne chelo la ya kari tamen tante tanzi chi yores.
All right, and then this is just highlighting, again, that there's the three different kinds of cells. We have what's probably an animal cell, and then we have the plant cell, and then also a bacterium.
Ta da de la ta pai ba nga chole thapong ka rik manda res phajo te ba na ane di karsa socha ki thapong che ane chajo te ba na dirwa zising ki thapong che chajo de ka res ne sumbu ki thapong res.
All right, so a little bit more reminders about the plasma membrane. This is showing the picture of the phospholipid bilayer structure, and you can see that there's water on either side, and this one is representing the inside of the cell, and so that would be the cytoplasm.
Ta da de ta pai ba na de la remo yo ba de ka res na thapong ki chimuti la ya nga su ge ko ramba ni tang den be ge karsa omi litsi se ramba ni den ge trio res gorwa ko ge chilo ta na lo ni chala ya chu yo res ta da de la remo de ta pai ba na de go wo la ya ta ja ju de ka res na ani wo de ta ramba de ni che go wo ke chu yo sa de ka res na ni thapong ga nang ti chog yo res thapong ga nang la ya thatsi yo wa de chong yo res.
And so I think that there was an activity you all got to do last year where you drew with the wax and then saw that the water went around. So if you recall doing that activity, that's a demonstration of this function of the plasma membrane.
Ta nanjelo la che chu che ku chi drio res thapong ge chimuti tri de tang ta chu na lo la dua tang chlo la dua ge so pa trio res te che ku ti tengi yo pa ba na ti kap se ge ani chu de nga chu thapong ge chimuti la ya shi sha de ta che ku ti drio res.
All right, so we've got the phospholipid bilayer, as I mentioned, and it's composed of two layers of fats, and it has hydrophilic, so that means it's this part that's going towards the water, so it has the hydrophilic part, and then it also has the hydrophobic part, which is here sort of essentially hiding from the water.
Ta da de ka su ta bai ba na ko la yo omi le sin ramba ni yo res chil ge ramba ni yo ba che ta te na la ya ta ba imba na ane ko tambo te la ya do do yo se te chula mi yo ar do chula ga ni se go ro chu ge shola kha chot ni ge ne go te ta chula ya ma ga ni chu ge chu ta chula jap te ni ge ne de la ya chu do ken ju ti yo res.
And then this is just kind of showing you in a larger form that up here would be the polar hydrophilic head, and then down here the non-polar hydrophobic tails. And it's related to this image, is just you can see how they're arranged to one another and the ways that they come together, that the tails are here together and then the heads are outside facing the water.
Ta tanda ge de la ta ba na phajo te ge mu nang tripcha chongcho chi chambu ju sha ju res ro ta de ba na go te ba na ta go go yo sa te chumi ge go ta ne wo te chula ta chu do kun ju ti cha go res ta phajo te la ya rimba te ta ba na ko ge ta chu gi do ke ju te ni cha nyam te koran ni khate sha duas te ne chu me ge go te ani chlo sa du ste de res.
All right, and then this is to remind us that when we're looking at the cell membrane, that phospholipid bilayer is not the only thing that is there. Within that layer, there are several other kinds of molecules that can be embedded that are important for carrying out the functions within the cell.
Ta nga su ge thapong ge chimu la ta ba na thapong ge chimu de ane omi le se niram chi oro ar ba ni cheng ge omi le se ti chi pu ge trpa ma res te ge na la ya ko ge thapong tha senda ten da yo res ro ko ge trpcha senda ten da yo res trpcha senda te la ten de te ane chimu ge cheli te drip ge chi yo res.
These main functions are going to be protection. You don't just want any kind of molecule going into the cell. Transportation, because you do want some molecules to be able to come into the cell, and then also for communication. And so this would apply to both unicellular and multicellular organisms. Either way, the cells need to be communicating with other cells.
Ta chi senda karyo res stambala ya ane kola tangpote la ya songcho gte jigu yo res ra thapung ko chimuti ge songcho jigu asena thapung nang la ya kanjo manjo ta yo marwa te goba jigu ro songcho te chigiche ani pati imba na ane ya ko ya beti kasana de ko yo marwa chlo ne nang lo la ya chi onden jigu yo ro as chlo ya ta nang lo ora jigu ro as te sa ba onne ge chele te yo res ta thamate la ya dande jigu yo res ta dande chi ya te imba na ko thapon chiden la yore thapon manden la yore sirwa ko chalone nalo la ya dati tangwa yore
Over here, this is a reminder of some of those different kinds of biomolecules. And I guess just to sort of put this into context, these biomolecules are important in the cellular membrane, and they're important in many other areas of biology as well.
Ta shende bana nga su de la ya biomolecules na chidengi dudu yorwa chidengi dudu kha chi nguto chi US de tshayus ta di kars ko re sena chiden dudu didake ani thapung ke chimuti trepa mase ani de la ya cheli ke chembu ndong yores.
This is showing the examples of some proteins that allow for transport here, and this is also saying that carbohydrates are in the plasma membrane to provide some structure. So it does need to be fluid to some extent, which is why it's called the fluid mosaic model. But of course we can't have it just so fluid that it's completely falling apart, and carbohydrates help have some structure.
Ta danda digane kola ya tripchata ba imba na tamputela nga su ke tzi ke tripchati yores tzi ke tripchati imba na ko chala ya ma ondin chala ya le phengi yores nibatela imba na carbohydrate yoro carbohydrate yo pati ke karchi ko res stambala ya ane kola ya chakko ke sheli chigdus khuto chigres ta chakko ke ti chigres stambala ya kola ya zuta chi jeng yo res ta ro zuta chi majim ba imba na ola itela nga so fluid mosaic model as to ro shertan thawda bi pisati imba na kola ya zuta mate ba imba na ko karsa ta nga so sherkhu ngula de pa imba na ko ga number ti chu chone tont kamarwa oti toela ya nga so ge carbohydrate ki cheli ti tong res ta mi tsinye mambo da ya jam da chi some are close in their eyes yeah.
I know when we talked about sleep up here earlier, I was like, oh, now I know. I got to know who to keep my eye on.
Yes.
All right, so we've been talking about the plasma membrane that's on the outside of the cell. Now we're going to switch to talking more about the inside of the cell and talking about the genes that are there.
Chapung target of Chetela is that and then Bananga Chapung Nalo Judete, Chapung Nane, that Trizita, Zuege, Chashichigorwa, Tetalatagians.
And I thought that a fun way to start thinking about genes would be to show you this picture, because this is actually a picture of my chromosomes.
That's it. Riziga kola motochis tabalaya, energy is Tamat Dembi on the Deshaja Chilaya, Perdi Tanito Sons, the Perdi Carresena, Perdi and Narangi Rizeti and Ani gets your ponte Kupati race.
So in case if it's a little bit small to see, they're all numbered. So chromosome 1 is here, chromosome 2 is here, and I have 2 copies, right, because I got one from my mom and one from my dad. They're numbered all the way to 22, since those are all of the autosomal chromosomes, and then there's also what would be the X or the Y chromosomes there.
Delaya Toya Kaputa, the Chonchon Dosta, de Taba in Bana, Neko Ponte, the La Tanka Jasha Doas, or Tanka Kosha Yoresta, Tamponi, Nua Sambashuachi Dos, that the Nustani Patokosha Doas, that it Chupon, you batting us Chacha Yungo or what you partner money on Gorwa, and the Tamading us with Samagu to Chagores.
So I have a question for you. There is something that just from looking at this, it just disappeared. No, okay. Just from looking at this, that there's something we would know is definitely different in the chromosomes from what we see here, since these are mine. And so there's something that would be different, Dawa compared to me, or Miguel compared to me, or compared to most of you. What So what do you think would be very obvious difference in our chromosomes?
ta tanda dela imbai na rowa dela yopate ngarangi chupong gopati res ta dela ta ba imbai na rowa ta dawai na digur ge mige na nu chechu manchi shi ta nchi mando yo sate kange insares. Say last one.
Yeah. The last one, exactly, right? So here is 2 X chromosomes, but then for biological males, then this would be an XY.
Ta came by narwa the XY yongorwa. Then the XY is for women, right? For female.
XY is male.
Okay.
Yeah.
Then the dim by narwa, then the delaya and X dengue race, Roanish, then the touches sama, Manchishilaya, Puins Tambalaya, and the XY chag race. Semigusuponde. Yes.
Okay, so this is another picture. These are not my chromosomes, which you'd be able to tell because this is an example of an XY chromosome.
Then they young question that is take Narangi Chopongi party maris, Delata by Bina, Deganalaya, Netama data by Bina, X and Y in Barua, the Mando in Bati, wants to book on the tongue to us.
And so when we're talking about the genome, that's talking about the entire set of the organism's DNA. So this is showing the genome of this human.
Tadanda de la yang atsu genome kecheons tambalaya mi kansa chila yopige chupon chatswa de la desijagores tatanda dig pandignalaya ane mi ta kansa chiki rizigi ta chini su chatswa chindena gogores.
Humans will have 23 pairs of chromosomes. This can be different in other different kinds of organisms. But what will be the same in all the different organisms would be this double helix structure of the DNA.
Ta karayore senaro ngatsu druami imbayna druami ge DNA na tabai bana chupon chan sasmi yores yang roa ta shenda la ya tabai bana yang chi jingwe dang so cha shenda la tabai bana yang kola ya chupon ge chate yang mi ta manda wa roa changate manda yong res ibana kola drabi cha karayore system bala de la ngatsu ge karsa juzuku ngo yo sa sto double helix roa juzuku ngo yo pate samala cha chung ga yores.
So what we're seeing here is basically it's zooming in. So here we're able to see the whole chromosome, and then now here we have zoomed in into this little segment where we're able to see a few of the different base pairs.
Nahanta tanda dela tabai bana tsushoko dela tabai bana ngatsuko chupong ta chanyu sasun kanza tago yorwas. Ne phajo dela ya ngatsu juzuko ngu dela roa. Juzuko ngu dela tas tambala ya chupong no tripcha chonchon chi ja chel de tane ngatsu tago yores.
This is if we were to go kind of zoomed in all the way and to see the exact sequence of that DNA represented by the A's, T's, G's, and C's.
Ta tanda de bana roa de kare system bala ya ko ge ta tripcha cheta pa chonchon te ja che de tan ko nana ya ane ko pa kanda do chi batela ta yores.
From the Human Genome Project, we do know the sequence for most of the human genome. So what I'm showing you here, this is truly the sequence of the very beginning of chromosome 1. So that would be, we're kind of looking here just at the tip of chromosome 1. This is the actual determined human DNA sequence.
Tanda dege chupong tampo dege ngoga tripcha chonchon chigite ku khatam de tane ko nangla ya tripcha kanada uchi pati resta de baina de ko the hume ngatsun drumi ge genome ge lecture te baina genome chama de ko go yoro kanada uchi me ro rizik tripcha karima ote tata de yores tanda tripcha chonchon chigite de res.
So it could be possible for me to make a lot of slides like this and actually go through and click and show you the entire sequence of the human genome. However, that would take about 6.3 million PowerPoint slides. So we are not going to do that. I will not make you sit through 6.3 million slides.
Tata dus de tam me genome na la yopi ge tripcha ti dus re la temba imbaina roati ten la ya ane ngasu ge lokto nyen ri ti karsa saya 6.3 last 6.3 right saya sumsa twitch google yores wa re la dus tan chi chemba bana ne tinano chi tan ding asu chi means.
Yeah, we'll just look at this one for an example.
Tante pitson some shiba, yes.
All right, so this, again, just a little bit of review because this is all going to come into play as we talk more about development. When we are looking at, this is showing us a central dogma here. We've got our DNA code here, and then transcription occurs where we have the mRNA copy of that, which is then going to be translated into the protein. And this is also just making the point, of course, that all of these are read in the triplet codon. So we read the base pairs 3 at a time.
Tanda de kare system ba nga chu sumsen sangda de kola res ta de chichu ko kwa leng yo pa na tawa nyang pu de res tawa nyang pu de wa tambo de la ya ane DNA ne ta da RNA la jura RNA ju de ten kar sa tambo phap su chi te ji la ya phap ju chi go yo res wa je tan te ko ge da te dia de la ya da te sumdi sumsen de yo res.
Okay, good. I was hoping it would come the other way. All right, so now we're going to talk about mutations, and I'm going to have this be something that you, again, discuss in your groups and just see if we can sort of collectively remember what we talked about from last year. And that is just going to be the different kinds of mutations, right? So there's several different ways that there can be a change to the DNA. And for now, I'll just have you all talk in your groups and try to come up with your list of what are the different kinds of mutations that are possible.
Ta tanda de ya yang she chula ya treated chi ga ko ka chi te ge yes ta te kare yin system bala ninjur go ko yin ro ninjur ko te nanjula che chu ga ja yo res ro ta tene tin system bala che chu treated chi de te ane ka chu chi tamba su go do te le thap chi na ro chi sta jodi kare yin sen na ninjur la ya ri ka chu yo res ta ninjur thep ta man ro ka chu chi yo res o te ko la ya treated na ro chi.
All right, thank you. Can we hear it now? I keep trying to, okay. Thank you everyone for your great discussion of mutations.
Yeah, that July apanang dos. To the pay apanang do tuche, Sataya Seronas.
Yeah, it sounded like everyone was very engaged in talking about mutations, which I appreciate. So for this, we'll hear from a couple of different groups. But again, I like to hear from different groups, so if your group decides to share, if you could just tell us about one type of mutation, that would be great.
Tatanda yang chiru komando mandone de kejes sononas din stambala ya chamala ya segorta mando ko achete ruka re ne ane ninju chi sona digdus
manzu guru de manzu 단다 제네디 아르나 팝시지그라 팝시지드나 이게 최소한으로 딘지송아 있나요? 전지만으로 그렇대. 인저때.
다 체바데 가르도 엘리미네이션도 가르도 deletion 체바데 deletion 왜 제주때 왜 deletion.
Yes, okay. And so just to clarify, is this, are you talking about there could be a deletion of
in the
one nucleotide?
Yes. Kone chigore
단다 니 제네니 아레니 팝시지그라 팝시지드나 디가 손다디노 이긴다고 직 최소화했나
그래
어디 기지
상다게 숨생는데 야치 체고로 와
야치 제
your code one code is missing or deleted.
Oh, are you saying a whole triplet codon, like a whole?
No, not whole triplet codon, just one single from the triplet.
Just one. Okay. So then what would happen if there's just one nucleotide that's been deleted? What's going to happen to the rest of it?
다 깨때 되나도 총바인 바이나 스와 다 가르쳐 그랬어.
단지베데 전지 직전거라 전지데 따다데 지기 답정 전지 만들어 정.
So we will so it will be transcripted or translated into a different protein.
Okay, yes. So everything after that will become different because if you delete one nucleotide, it's going to shift everything over. Yeah.
다 단데 디나네 고 다치기투스 게 메퍼탕 바인 바네 체파인 바나스 다til레테네 데데 땅아 속에 디치고로와 드지라 팝지치고로와 팝지치고 값에 디기 질라 드치고 드지 참마 만들어 차그레스. 다 젬바송다 더더더 지기 요바단다.
네보르샤기 만조디나. 네보아디.
네 포대 가르
transcription location. Transcription location. Translocation.
Translocation.
Could you just describe what you mean by that? Like what would happen if there's translocation?
Translocation से ना कर सको रेसा
Such as
अले कोडन ची चे देते या ताँ इधर है
ship.
Pena dini da jara ACG na jidin thama de na changse jid khang le thung re jo
कल लगवा frame shift लगवा है
dam da re ta
yeah frame shift mutation
I see. Okay, yeah. And so I think that would be similar to what we're talking about before then, is that way if you have deletion of one nucleotide, you have a frame shift because everything else moves over.
हाँ तंदा दे गांव दिला या deletion तो दो शक दो स्टार्टिंग स्टम्बला या कोई नानी nucleotide चीज़ से करो आता न्यान्दु के टुच्चा चीज़ ती मैं प्रताम बाइन बाइन ना तिला तिने देते हैं अन्य ड्रम कांसा या योंगर वास okay
pena ni zu cha de ra e na de tha de den do samso de dini ya thung ro jo ra de jo ra ya move che jo ra de la go amar da delete do go amar da ya de na ne pone desa sa je jiblo ne
the
dragin je
they call it translocation right so he's saying like if we a at the end sometimes it changes its place to go to the front and then we have a different so he's saying that it's not like a deletion but
Oh, you see. Okay, so I think that typically will happen with larger sections of chromosomes. Like that, yeah, that won't happen so commonly with just like this little piece that we're looking at now. But yeah, sometimes it will happen like a whole piece of the chromosome. Weird things can happen. Yeah, that it kind of comes and then it gets flipped around like that. So okay, yes, I know that would be like a big scale chromosomal mutation.
हाँ तो तंदा ते बना गा सुपंग करने को ट्रिप चे छोंचों से माइन बात ट्रिप चे चेंबू मुने सकते हो को फ्लिप चे और सकते हो को सच्चे जीव जाबले और तीन चोंग रेस
Any other kinds?
Injure. Genome injure. Genometic mutation.
Genometic?
Mutation.
Genometic mutation?
I think I would need more of an explanation of what that means.
कला को रेसा
The several of town you are ready.
changes in the numbers of chromosomes.
Oh, I see. Yes, yes. Okay, yeah. So this can also happen at the chromosome level, right? And so usually that would be probably resulting in an organism that can't live most of the time. There's only a few cases where an organism could live with different, or this is for humans, only a few cases where humans can live with different number of chromosomes. And yeah, usually that would be a mutation that means that organism cannot survive.
तंदा के genome के निंजला छुपा इम्बाइना करी शकरे से ना तको छुपंगो ngola या निंजर थे गयो चांस ता छुपंगो ngola निंजर थे से छुपंगो काटन ला निंजर थे गदोआ इस तंबला नेतां देख का से इम्बाइना चिरगे ना ता चिंगवे ते छुने चिया खाबु चिगरेस मानशिशिलाया ने खाशिलाया को छुने चिया योंगर बंदा ने मानशिशि को दिनारो निंजर थे इम्बाइना छुने चिया खाबु चंगरेस
Yeah, I just was thinking of it. The only ones I know of in humans where they could potentially live with the different numbers of chromosomes, it can happen, I think, for chromosome 18 and also for 21. So something that you might notice, right, is those are smaller, so they would have less DNA.
When you say 18 and 21...
Oh, and also actually the X and Y also, yeah.
Okay,
I'll just re-say. It could be possible for humans if they have an extra chromosome so they could have an extra chromosome number 18.
Okay.
An extra for 21, and also of the XY.
Okay. That under day nane kosuke genome ki ninjer sayati imbaina samlaya ngasuke chupon te thepa upaina ya nit ko dromila se korwa chupon thepa imbaina ko nit khatina jongores ta thepa yoati karsena so te ko ge tada de kap se tapa imbaina chupon ge dela tapa imbaina ko chupon te jenda ta durba imbaina te chonchon rdoa 어때라 때문에 됐다 용 거레스 다 피차샤판에 대파 용한테 내탄 카시치 용 거레스 다 담보데 임바이나 조제티로와 조제티라야 대파 용나용 코니트 거레더니 뉴스사치 대파 용스 바르스도가 되게 제라 뉴스사스 바데가 소그 재미그 초본데가 대파 용스 바레스
Let's hear maybe one more idea for, let's kind of focus on a nucleotide, like not the big chromosome. We can talk about a nucleotide change, yeah?
다 초pon가 개체마셔지 되든가서 원사가 듭체 걸어셔 봐요. 마이나 춘춘기 테니셔 봐요. 바나 젠다 뉴인지를 가려래사.
단다디 상다드소디 다 숨숨 열어. 디나로 숨나로 양 튀어바지게 열려와네. 디니 디 월에 차상 마트시 개발라 때네 다 냥가 치셔디 단다 스톱 사인 열어. 딜로 주와테스나 닝아서 캔서 나야다 펫 셀렉게버 튼샤에 줘라. 까마토에 차그레스.
Data insertion 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 mutation is there like called insertion mutation. If you insert one nucleotide at a point, then pretty much everything will change. And sometimes we have sometimes in some cases we have stop codon where it shouldn't be, so it will mess up the gene sequence.
Yeah, exactly. That one I could get from the stop sign. I was like, I know what that one is.
Tanda delaya yang ko ge gela songati baina tanda delaya ko sam jogage ko sangdachi ngudwa o te tanda ngoto chigins.
So just to sort of quickly review the types of mutations we just talked about, as well as kind of adding some vocabulary terms into there. What we're starting with here, this is just the normal, this is a DNA sequence that does not have the mutation. And just what we're seeing here is that we have both strands of the DNA. So you can tell that these nucleotides match up, that the A is with the T and the C is with the G. Then we have the mRNA here, and we also have the peptide. So this is showing the different amino acids that would be in the protein.
Tanda dela tabaimbana tanda dij de tanga su kare tagin sena ane ninjurge ta rikare yopate khachila tagin sro tanda dela tabaimbana gala peso yopate ninjur the mepege ane ngujire sro ta tambotela ngasuge DNA ge koge ta te nyama song yo ro ni cha te tambo nyo te chag yo res ro ta tabaimbana e tang tri tang e ta sita ji rig do akora da digate to sungor wa tigi walk this smati kare system bala ngachu panchen RNA ngala kariyo patires ro tigi wo te kare system bala tetala tene te ngachu ke tiziti kare tukores amino tiziti kare tukores te ola te yungores.
This is one of the types of mutations that I know some of the groups talked about, but I actually don't think came up in the discussion, is that sometimes there could be what's considered a silent change. So it's possible for the DNA to change, but that there is no change to the protein. And just one thing that's important about this one is that you can see it here, that it's the third base in this codon, that it would be usually just only this third base could change and result in a silent mutation. If it's one of the first two in the codon, then that would be changing the protein.
Ta tanda dimba na tampote kare system balaya ane tirwa ane ngasu kusumbi ninjir deres ro ta dikarsa silent mutation sigdo kusumbi ninjir deres ta dikarsa koresena tanda digina ne so de sumsang na tamatela juran dugo yores ro sumsente tama juran dugo yore ko mando chilhego ta chapchi chigdo ro sabchi go ta imbaina tamatela ngasu goyage amino chirjila juran dugo resna dugo yo mare ro ta ngasulaya keti srota te sumchen nang tampo nicha te la yan juan duwa imbaina ane tila tene tripi amino chirjila juan dugres.
Another type here that I guess I don't know if we talked about this one either actually, but it's changing the amino acid, but it's different from that insertion or deletion where it's going to change everything afterwards. This one, it's only changing one amino acid in the whole protein. So it might have an effect, but it's also possible it might not.
Ta sendachi kare system balaya ane dignane amino chirji kan chigte juan dugo yores ro amino chirji kan chigte juan dugo yores ta tin system balaya delaya mi sense ngasu ko peche ton tril do triosans ro ton triba don't look like that, or don't look at it. The thing on a question you are letting the day, amino church constant you do your rest. And a thing by the young co sturgish kitchen chamber take your mares.
And then this one we already just mentioned about the stop sign. So some of the codons mean that the protein will stop translating. So that's an example here. That so in this case, it made the beginning of the protein, but now it reached a stop codon, so it's just not going to make the rest of it.
That's Sansamalaya, Kariongores, Samji the Ungorsta, nonsense that Stambalaya, Tom Mebara, Tom Chawata Yungoresta, Tion Stamba, Karachigoresena, Parotila Chipalatinidete, and you could get Dang agenda Chikasuten, they thought on the Pisha Shapana, Tangi del Sonage, could some shall say that it angores, Tion Stambalati, what is Homaku Mando Chagorwa, some shall and some Cheki or Roko.
And then the last one is the one that I think we talked about the most, and that's the frame shift. And this could happen when there is a deletion or an insertion, either way, because both of them is going to then shift the nucleotides for the whole rest of the DNA, the reading frame of those. So then all of those amino acids will be different.
Domjurti Ungores, Domtin Juraton Gotiores, that Tion pickups a Karchigores and a Chinjala Tonala Tendis, that day, Domjurdi, Nitanjagon Ungores, Chichibaina, Chualatin Ungores, Chichibana, Nane, Labalatin Ungores. That Domjurdi Testambalaya, Tigi, Gila Yopigi, Amino Church, Samalan Juartegiores.
So these are mostly organized in sort of how much of a bad effect they would have on the organism. I would note, I think my personal thought is, depending on the protein, both of these could be pretty bad. Like if it's an essential protein, a protein essential for life, both of these would be very disruptive.
That and the delay upon it with the cook shooting its subchilla and upon to say your rest, yo team by Benapet Duchachi to your rest, Imbanaya, Stirgicha by Manayanko, Tizilla Raligores, Katie. Tiziti any Churila Hachi Kitchen within the Yopambana, Okan then Chime by Nayanko, Kartin, Injurtem by Bana, Tilatin the shooting and by Shuchambu Grace.
All right, some of this we've already talked about a little bit, consequences of mutations. And there's some pictures there, right, showing this mouse that has the different changes in coloration. And this is to be a reminder that mutations can occasionally be beneficial, such as in this case, if the mouse is more camouflaged in its environment.
The Dicary system balay, Ninjula Tenedatan, Driva Kanadu Chungu Metres, the Dina Netaba in Bana, and some Laya, Ninjula Tenedatan, who keep in Waganatan Gores, that under Dilayopi, Cicilla Tapa in Bana, Korangi, not in Jutkudua, not in Jutku Patilla Tenedatan, Kola keep in Ragores.
Okay, so oftentimes if we're talking about these beneficial mutations, we would consider it to be an adaptation, which came up in our characteristics of life. And this question is what we'll explore in another quick activity after this, which is just what is going to determine if a mutation is beneficial.
Ta tangi dena la yang su chesho ke karsa chideng gi chechu suwa na sing gi chechu ge tunjur te segores ta tunjur se ba imbana ani keti ninjur chi the de te ninjur te la ten de te campaign chung go pa na nga su tunjur sa ten de se ja go res ra ta troate kare system ba chesula troate kare system ba la ya ninjur chi campaign dem bare se te kandas ta che gres kare gi ta che gres.
So now we are going to go through a little reminder of the peppered moth example in case you either have not heard it before or haven't heard it in a while. And as we go through, I would like for you to be thinking about what would your hypothesis be for what could be going on with the genetics of these moths?
Ta dene tanda dete sheja stambalaya ane pep karsanga su simbutrajis lembuchiku gola chimalechiku gola ngasu du stayo res roa ta tila taba imbaina ane kochula ya ninjur thebala tinedete tandela ya tunachi ate karses tamba ninjur chebala tinedete kochula ya khipen kari choma chongla sobati gola ta go res.
All right, so just as a reminder, these are what peppered moths look like. And I'm curious about this. I want to do this by a show of hands. And so this is just totally your opinion. There's like not a right or wrong answer to this, but can you raise your hand if you think, so these are the two options. If you think this moth is more camouflaged, the darker moth is more camouflaged.
다 단다들라 야 저쪽 지금 삼슬라 야 삼슬 들어왔으면 yes 라 리턴 너요 말이스 다 단단데 나네 그게 델라야 그 숨버터지니 도와라 숨버터지니 도와 뭐니 요레스 단 되게 나네 다 camouflage system 가서 돈지라지 거와 코리라테는 돈지라지 거와 내가 나네 가마 델라야 코리라테는 돈지라게 네탄테 슈치원도 세바나 라바창로지 에사
Yes, we can do a hand raise if you think that one. You don't really think this one? So does everyone think this one's more camouflaged?
그때 안에 그 코리라테는 돈지라시 요란 돈지라 네탄 슈치원도스는 라바창로지 에사
Okay, so it seems like a lot of you are thinking this one, but maybe a little bit iffy either way.
다 만시시 오그텔라야 군돈지로게 뉴바티시 치어두세 데샤스 네 카시시게 타고 강게나디도 강게데렌도 상구두스
So the idea for these is that these moths are on the trees in pre-industrial time, so this would be before there were factories and industries that were creating a lot of pollution. This was sort of the original state, is that these moths were living on these trees, and they were generally similar in their amount of camouflage.
that there Tambalaya, both of them are in pre-industrial, right? That and then the Tambalaya Nichage Neta Nicha in Bananga Suge Zunichi, Zurichura, Zurichura, Turku Netanchurwata, Zutagri Matergunga, your is the Koran Chain Bana. Kori Latin data Netan Denado, which Juradus Tutku.
Okay, so then this is just showing after that time what happened, right? There's a lot of factories, they're creating all this pollution, and so then that's going to cause the trees to become a darker color.
Ta ngongete ko de zoli de mater gungona che tige jela ya zoli de tan sambala ya longkang ga baso shi chambo che wala ten de ten ko ta shindong ge chi shi ge dote ane this do napo cha ge yores.
And so this is, so we have the before picture and this is the after picture. And I think here it's very clear which one is more camouflaged.
Ta de kare se stambala ya ko buti ngongete chiba chichanche ane ko ge ta baso khori baso chi wala ten de ta chonran jusha yorwa tela ten de ta nga sunde ta ba na gangi chi ane ko khandon jira chi gorwa tig niwa shi chambo yo pati ngosu do budu as tig gan ta ba ina.
Yeah, and so if there's some birds and they're looking for some food, which one is the bird going to be most likely to eat?
Ta kete isro ta tanda netan dena do kaps ni chagi ku ge khasi chogu yo pa na digi nang ni kange chi chai gi sasares. This sasara. Yes.
Yeah,
this one.
exactly. So the birds are going to be way more likely to see that one because it stands out a lot. So when they're going over here to select their meal, it's a lot more likely that they're going to go eat that moth.
Ta ko ge jalbak ke khoi la ten de tan dan lo la ya ta ngosu to pakari men de sagarwas tis tambala ya tan da digi netan kaps ane chi tis karkai ba tis sagres.
Okay, we're nearly at time, so this is going to have to be a very short discussion, probably only 2 minutes in your teams. But come up with what you think is a hypothesis for what could have happened to the genes of that moth. And so there could be a lot of correct answers, but just some idea of what might have happened to the genes or the DNA in that moth.
Ta tanda de kaus che chu ge Twitter giants Twitter ha chang tin thong thong kama ni ga na te Twitter giants ro ta Twitter de kare se stambala ya sumbu thari de ge reji na la ya karin jong go yo res te ko la ya chojak ji tro che sa Kusa tanda which do a budigis reji na la ya ani kari chung go yo res tanda netan de kaps te ko la chojak ji tro che sa
Oh, I didn't get to the affection. Okay. Okay, I love all the discussion, and I want to make sure we get to lunch on time, so we'll have to wrap up.
Ta dulang yapu tude pe apu nang deshas. Ena ngasu nyungkon shelala tata pegu dus din stambalaya. Ane dus ta tude dus samcharo chis.
Great, so let's hear ideas from 3 teams since we're trying to go a little bit quickly, and then we will get to lunch. So 3 ideas of what possibly could have happened to the DNA.
Ta tanda de la ya chopo mando asum ro roko sum gane dus ngasun jobo dus nyenchi de jela ne percentages.
So the reaching one yora. Carbo reaching one te anekarasina genie stone, no power reaching one yora. Hotel Rapshama Jeton Genico Rigitil and Gerati Grace. Tomitom Carbo no Ponigura.
Reaching one take a prototype.
Eller. Reaching one chassis or chassis, reaching chassis.
Phenotype reaching one the phenotype So the hypothesis is that the phenotype for the black moth will transfer to the next generations, but not white.
Okay, so we're thinking about the phenotype and then relating that to what the genotype would be, the DNA, is that I think you're saying what would happen, like genetic changes here would be that the genes of the black moth get passed on and not those from the white.
Ta tanda denga so rizing ono la tapa imbana ta rizigi chechula tapa imbana ta de ka karsu system balaya ane chi ta nakpu imbati tan devi rizite jimal rachmala khutra chepatan karpu imbati ramjing chadwa tisgorwas. Yes.
Nganzu chena rizil ngyo rogu maris.
So we think there won't be change in the genetics.
So do you mean the DNA of the black moths won't be changed, or?
Karila dugumar segure, karpute segure, nakpute segure.
There is a nabudu mutun soneshi to gora. Kuruki chenchi amado korangi rizil and joan joja ginsen soneshiba maris.
So with for both cases there won't be any genetic changes. It's only because of the environment that the black moth will survive and the white moth won't survive.
Okay, I see. Yes. Okay, I understand what you're saying. Yeah. So right, there's not going to be some like special mutation that happens to come save them or something. It's just now their DNA is how it is, but then the white moths will more often be eaten. So yeah.
Ratanda de bana ko korila tende de tero ko rigilan joran du mares korila tende de tani karputi mans ko suge sai chinyon du res naputi bana ko suni chigrama to pe rigilan joran chigan du mares.
Any last different ideas?
Te ta mandra ge an samse jemba tinda ayo sa.
Okay, enjoy your lunch and we will see you this afternoon.
Ane shela tu na